The third part I will mention here is about host-targeted inhibitors. First, we should ask the question, why we need host-targeted inhibitors? On the right part is host targeting. On the left part is the virus targeting. The advantage of a virus targeting inhibitor is it's very specific. Specificity means less toxicity. However, it has a major limitation, which is it's relatively easy for virus to generate mutants. If we look at the host targeting inhibitors, normally it should be broad spectrum. Well, in the sacrifice of higher toxicity in some case, a huge advantage is it's usually difficult for virus to generate mutants because it's the cell host target. And in some cases, it can be used as orphan disease treatment. The first host target I will mention here is DHODH. DHODH represents dihydroorotate dehydrogenase. It works this way. DHODH can transform dihydroorotate into orotate. The orotate will be further transformed into the UMP and then UDP, UTP, further involved in RNA synthesis. Therefore, if we can stop the activity of DHODH, that means theoretically we can block the virus RNA synthesis. DHODH lie inside the mitochondria. Last year, our own group has reported discovering a new class of chemicals as DHODH inhibitors. On the left part, we can see the structure of a representative chemical named RYL634. We have found that this type of chemicals are highly effective against a broad range of RNA viruses, such as dengue, Zika, EV71, RSV, and MERS coronavirus. In the early of this year, a group from China also reported developing DHODH inhibitors as potential coronavirus inhibitor. We can see their data here. In this table, we can see the compound S416 is very effective against the coronavirus. It has the EC50 value around 17 nanomolar. It's the best effective compound so far as I know. So I'm pretty anticipate the compound in the near future. There's also a drug candidate for inflammation has been proposed for COVID-19 treatment. As we know, cytokine storm can cause serious symptoms for patients. And those serious symptoms has been found to target as the host cell. Here we see the first drug, thalidomide. It has been used in clinical trial for anti-inflammatory treatment. Thalidomide can suppress virus replication. Also, it was believed it can suppress overactivated NF-kappa B and suppress pro-inflammatory cytokine. Due to this reason, it can be used as anti-inflammatory drug, as well as immunoregulator. On this figure, we can see thalidomide can lower down the TNF-alpha amount. This table shows the inflammatory cytokine and the lymphoma site in a patient. As you can see in the table A, thalidomide can decrease the amount of inflammatory cytokine in patient serum. And in the table, in the figure B, the lymphocyte in serum get increased by using thalidomide. The second drug I will mention here is named chloroquine. It's a very famous drug. Chloroquine and the related hydroxychloroquine, both of them are originally developed for treating malaria infection. In this paper, chloroquine has been discussed for its detailed mechanism. Actually, the true mechanism is quite complicated. It has been reported with different potential mechanisms, such as acidifying the lysosome. Chloroquine may presumptively inhibit capsaicin. It may inhibit different kinase, interferon M protein, alter variant assembly in the body. It was also reported that chloroquine can reduce the production of pro-inflammatory cytokine and disrupt viral S protein. In the early of this year, there's a Chinese group has published a paper in Cell Research. In their report, they found that chloroquine can demonstrate a significant in vitro activity against novel coronavirus. You can see here, it has a EC50 value around 1 micromolar. Based on their immunofluorescence microscope study, the chloroquine was found to effectively inhibit virus infection. Besides chloroquine, in the paper, they also proposed that two other conventional drugs, usually it was developed as antiemetics, can also demonstrate inhibitory effect against novel coronavirus. For example, like this compound, it can against coronavirus with a 0.2 micromolar activity. Here we see the structure of the niclosamide. Last year, in the Journal of Nature Communication, there's a group report their discovery about the drug mechanism. They found that this drug can inhibit SKP2, enhance autophagy. Therefore, it can stop the MERS coronavirus replication. Then it was proposed that this drug may work as a broad-spectrum antivirus. On this slide, we can see two drugs named chemostat and nafamostat. Here's the chemical structure. As we know, novel coronavirus use ACE2 for entry, and the serine protease TMPRSS2 for spike protein priming. And those two drugs are believed to be TMPRSS2 inhibitors. Then as a result, it can block coronavirus spike protein-driven entry into the host cell. On this slide, we can see a figure shows that chemostat can inhibit the coronavirus entry. This is another drug named omeprazole. It's originally developed as a proton pump inhibitor. In a recent preprint paper in website, it was proposed that the omeprazole can increase the anti-coronavirus activity of remdesivir and aprocitentan. Here is the curve. Besides the above-mentioned drugs, people also proposed that chemotherapy drugs can also help to fight against the coronavirus. The strategy is like this: to increase endogenous Mg. Mg means methylglyoxals. As we know that Mg modification can inactivate the virus. Here we can see 3 proposed chemotherapy drugs, such as BBGD, doxorubicin. And those 3 drugs can increase about 4-fold Mg in HEK293 cell. And in this preprint paper, it was proposed that those 3 drugs may help elevate the symptom of the COVID-19.